evening. Today we are going to be introduced with a new chapter of biology which is not only interesting but also very much useful in our day-to-day life, and that is nothing but biotechnology and its application. So first let us know what is biotechnology. It is the technology that utilizes the biological system. And in today's era, in this 21st century, this biotechnology is very much helpful in our day-to-day life as it deals with the industrial scale production of different biologicals, which include the biopharmaceuticals, means different medicines, drugs, etc., bioremediation, the waste management, energy production, etc. So first, let us introduce what is the usage of biotechnology in the field of agriculture. So India, it is a developing country, and being a developing country, the 20% or more than 20% of the country's GDP, it comes from agriculture, and more than 50% Indians are associated with agriculture. So it is very much important if we can do something to develop or to increase the yield in agriculture with the usage of this biotechnology, and even without causing any harm to our environment, then that will be very much beneficial for the mankind. So let us know first of all what are the advantages of using biotechnology in agriculture. So the first thing is that it makes the insect resistant plant. So it makes the plant stress resistant. What about the stress resistance? Is it like any kind of stress that we sometimes go through? No, it's not at all. The stress in case of plants, this is nothing but the stress like the biotic stress, like the pathogen, the plant pathogens like various bacteria or fungi, or sometimes the abiotic stresses like the climatic conditions, severe climatic conditions, a very hot weather or very cold weather, or extreme drought situation, or salinity, that means the excessive salt in the soil, etc. The second advantage is that we don't have to use any kind of harmful chemicals like insecticides, like pesticides, etc. So in that case, the natural fertility of the soil is not lost. And in this way, we have to make, or we can make, using the technology in the biological system, those are named as, or those are termed as GMO, that is genetically modified organism. GMO, that is genetically modified organism. What about that? What about the genetical modification? Genetical modification of any organism, that means the manipulation of the genetic material or the gene of any organism, that can be any plant, that can be any microorganism, by ourselves, by the biologists. Okay. Now, why will we do that? Let us understand this thing with the help of an example. The very common example, it is the golden rice. It is the golden rice. Now, it is very interesting, right? Golden rice. Here, let us see this picture. This is the golden rice. We can see here, this one is the golden rice, and this is golden in color. So, from where the golden color comes, and why, and for what is the significance of this golden color? The significance of this golden color, it is nothing but due to the presence of a chemical that is termed as carotene. I think this term, it is, we are more or less familiar with this word carotene. Okay, carotene, it is the precursor of vitamin A. And why is vitamin A helpful for? Vitamin A is beneficial for our eyesight. Okay, now India, like a country, like a developing country, India, where the rice is the prime food, if we can eat the carotene or the precursor of vitamin A along with the rice, then obviously apart from getting nutrition, it will also be very much good for our eyesight. Now now this carotene can be synthesized by a bacteria. And the biologists then thought that if that particular gene that can be isolated from the bacteria and somehow if it can be inserted within the plant genome, suppose this one is the plant genome. Suppose this one is the plant genome and this particular gene, this I am drawing in the red color, this particular gene, it has been isolated from the bacterium which is responsible for synthesizing carotene. And now we can say that the plant genome, it has been modified. We can say that the plant genome has been modified and now it can produce this can produce the rice plant, it can produce carotene. So obviously the rice which will be produced by these plants, this will be increased in yield, there will be increase in yield also along with the synthesis of carotene. So this golden rice, it is an example of genetically modified organism or GMO. Let us know about another thing where we can use the biotechnology. So here we can tell about the pest resistant plant. We can tell about the pest resistant plant. Okay. So what about the pest resistant plant? Now there is a bacteria. The name of the bacteria is Bacillus thuringiensis. The name of the bacteria, it is Bacillus thuringiensis. And this bacteria, it has a gene, particular gene. What is the name of that gene? Cry gene. The name of the gene is Cry gene. Okay. This Cry gene, sometimes it is Cry1AB or Cry2AC, this kind of gene, they can produce a particular toxin proteins in the crystallized form. Okay. So these Bacillus thuringiensis, they produce with the help of this Cry gene, they produce a toxin that is very much lethal, that is harmful for some specific classes of insect pests, and those are the Lepidopterans, Coleopterans and Dipterans, like beetles like cotton bollworm, armyworm, normal fly or mosquito, etc. What happens? What does this gene do? Cry gene, it actually let the insects cry. Okay, we can think like that. Actually, the protein, the toxin protein, it is formed in the inactive form. So here also can come a question that why, if the bacteria they themselves are producing the toxin, then why the bacteria are not died themselves? Because the protein which they are producing, that will be remained in the inactive form within the bacteria. In the inactive form within the bacteria. But when this protein will be ingested by the insect pest, then they will be solubilized. How will they be solubilized? With the alkaline pH, with the basic pH of the insect gut. And after that, when they will be converted into their active form, after that they will be bound to the midgut epithelial tissue of the insects, and there will be created some pores, and ultimately the entire body of the insect will be swollen up and they will die. So this is the mechanism how the Bacillus thuringiensis bacteria, with the help of their Cry gene, they are making a toxic proteins, and ultimately that is lethal to the insect pest. And in this way we have already made some kind of insect pest resistant plant. Those are the BT cotton, BT tomato, BT brinjal, etc. So let us see some of the plants which have been made. See here, this one is the cotton plant, the insect pest resistant BT cotton. This one is the corn resistant, corn borer resistant corn plant. This one is the BT potato. This one is the BT tomato. Okay. Next, the next thing is that there is another mechanism. There is another mechanism which we can also use, and that mechanism, it is known as RNA interference. It is known as RNA interference. Okay, so RNA interference, it can be written as RNAi also. So what about the RNA interference? Interference means obviously the function of the RNA. You know what is the function of RNA? I think you have already studied it in the previous chapter. The function of RNA to make proteins by translation. By the process of translation, mRNA, particularly mRNA, within our cell there are 3 types of RNA. We know that mRNA, rRNA and tRNA, and among these 3 types of RNA, the mRNA, with the help of the process translation, they can make some specific proteins. And proteins means specifically enzymes. They will help in some particular significant physiological functions. And if we cannot produce the protein, then obviously that physiological function, that physiological activity will be interrupted, and that will cause a lethal thing, or that will cause a death to that pest. Now, which pest in this way they are killed? The name of the pest, it is Meloidigyne incognita. This one is the pest Meloidigyne incognita, and the host plant for this pest, it is this tobacco plants. So Meloidigyne incognita, this is actually a nematode. This is actually a nematode. Okay, what does it do? It actually affects the root of the tobacco plant, and ultimately it causes the reducing in the yield of that tobacco plant. Okay, now what we have to do? Now the RNA, if the RNA of this nematode pest, it is interrupted, it is silenced. Okay, then obviously it will not be able to produce the protein anymore. It is known as the mRNA silencing. Actually, this mechanism is present in all the eukaryotic cells. This mechanism is present in all the eukaryotic cells as a kind of cellular defense, okay, against any kind of harm. Now, the thing is that a double-stranded RNA, it is introduced from some outer source. A double-stranded RNA, it is introduced from some outer source like RNA virus, or it can be a transposon also. And with the help of a vector, that is known as the agrobacterium vector. So this vector, it is carrying the gene of our interest. With the help of agrobacterium vector, the outer source double-stranded molecule, it is introduced to the pest, and ultimately in their body, the RNA, which is actually a single-stranded molecule, it will not be there. There will be double-stranded RNA, one with the sense strand, another with the antisense strand. Sense strand, that means it is the coding strand, and antisense strand, that means it is exactly the same of the template strand which was used as the template in the DNA, that strand. So if there are double-stranded RNA molecule, then obviously they will not be able to synthesize the protein which is important for their survival, for the survival of the nematode Meloidigyne incognita. And here you can see that this is the plant root, this is the tobacco plant's root, which are infected with the Meloidigyne incognita. And in this way, by this mechanism of RNA interference, we can silence the RNA, that is known as the RNA interference, and ultimately the nematode will not be able to produce any important protein. So we can understand now, with the help of biotechnology, by genetic engineering, like using some important technique like RNA interference, or using some important gene like the cry gene of the bacteria Bacillus thuringiensis, we can develop a large scale production of yield in agriculture even without causing any harm to the environment. So that is all about the usage of biotechnology at this phase in agriculture. Thank you.